All right, welcome to today's 26th annual Karen Wetterhahn Symposium. My name's Holly Taylor. I'm the Associate Director of Undergraduate Advising and Research, and also the Women in Science Program Advisor. The symposium is named in honor of Karen Wetterhahn, Professor of Chemistry at Dartmouth. She co-founded the Women in Science Program and mentored many undergraduates who did research under her watch. Karen's love of science and desire to see more undergraduates have the opportunity to participate in science research led to the idea of a symposium in her honor. Today we celebrate this spirit and all of the undergraduates who participated in science research this academic year. We are particularly excited today to welcome Dr. Kyle Vanderlick, our keynote speaker, to campus as we celebrate the 150th anniversary of the Thayer School of Engineering. But before we hear from Dr. Vanderlick, we have some student awards to give. The first award is for library research in the sciences. I would like to introduce Dean Sue Mayer, Dean of Libraries, to announce this winner.
Thank you very much. I'm absolutely delighted to be here today to present the third Library Research Award in the Sciences. The award recognizes students that demonstrate an exceptional ability to conduct research in the literature of their field and who have developed an outstanding pattern of research and inquiry. The award is sponsored by the Friends of Dartmouth College Library and evaluated by a panel of librarians from Kresge Physical Sciences and Dana Biomedical Libraries. We received several outstanding submissions this year and are pleased to announce that the winner of the Library Research Award in the Sciences 2017 are James Howe, the 6th, class of 2017, and Jessica Kobza, class of 2020. James's submission essay was notable for connecting his literature review to scientific process, as well as for its extensive bibliography reflecting diverse sources. Jessica, who is also a Women in Science Program intern, clearly stood out for her sophisticated research strategies and techniques. Please join us in congratulating James and Jessica for their achievements.
They're here, could they come forward? I'd like to now introduce Professor Eric Fossum, Professor of Engineering and Associate Provost of Entrepreneurship and Technology Transfer. Professor Fossum is a recent recipient of the Queen Elizabeth Prize, and he will be introducing today's keynote speaker.
The introduction of the introduction. That's interesting. So anyway, it's my pleasure to be here today on behalf of the Thayer School of Engineering and all of you to introduce today's speaker. But I had to mention that my third daughter just graduated from Oberlin Monday. It's my third daughter that's also gone into science, which I don't completely understand. If you knew them when they were young and growing up, you'd understand what I was just saying. But anyway, it's nice to see women in science being honored and promoted. It's my pleasure to introduce Kyle Vanderlick, who is the Dean of the School of Engineering and Applied Science at Yale University and the Thomas E. Golden Jr. Professor of Engineering. She received her BS in '81 and MS in '83 degrees in chemical engineering for RPI. And speaking of Oberlin, I just drove through Troy on
Beautiful Troy.
My way back. Yes. Lots of traffic there, by the way. And her PhD in 1988 from the University of Minnesota, where she had a fellowship with the American Association of University Women. So after 1 year NATO postdoctoral fellowship at the University of Mainz in Germany, she joined the faculty in chemical engineering at the University of Pennsylvania in 1989. In 1998, she moved to Princeton University and became chair of the Department of Chemical Engineering in 2004. And in January of 2008, she took the helm as Dean of Engineering at Yale University, which, by the way, is where I did my graduate work, years before, I must say. But it's great, and I've also had a chance to work with Kyle recently, and as part of her advisory council at the Yale School of Engineering and Applied Science. So noted for her research in interfacial phenomena, currently centered on biological and synthetic membrane materials. She received the Presidential Young Investigator Award in 1989 and the prestigious David and Lucille Packard Fellowship in 1991. She's also the recipient of numerous teaching awards. So as a Dean of Engineering, she led the establishment of the new Yale School of Engineering and Applied Science, which, by the way, that school has gone through a number of iterations over its lifetime. So it's the new Yale School of Engineering and Applied Science in many ways, and has really helped shape the direction of the school over the past almost 10 years now. So I'm very pleased to welcome, I'll say, my friend Kyle to come talk today on retrofitting membranes and wrapping up where nature left off. Welcome, Kyle.
Thank you, Eric, for that wonderful introduction. Thank you all for coming today. Congratulations to all of you. You are, what, days until graduation? When, a week, 2 weeks? Can you feel it? Is it close enough? You're getting itchy. Well, congratulations. You've survived 98% of the year. So it's a real thrill and honor for me to come to Dartmouth. I've grown to know Dartmouth over the years. I've come to Thayer on a couple of occasions. And I often say to myself, damn, if I was smart enough at 16, I should have chosen Dartmouth to come. It would just have been a perfect fit for me, as I'm sure it is for you. So it's just a real pleasure for me to be here, especially on this wonderful occasion. I wish we had something like this at Yale. Maybe I'll start it when we go back. So I thought I would talk about a long-term research direction that I've pursued really for now almost 20 years, sort of with my left hand. So when I graduated in chemical engineering, took a chemical engineering curriculum, it was to avoid biology, okay? So biology was never a required class. There was really nothing quantitative about biology, nothing that was modern about it. Really, the revolution all happened once I graduated with the advent of molecular biology techniques and so on. And so biology was just something, I could spell the word and that was it, dissecting frogs, that kind of thing. And when I started my academic career at Penn, within about, I would say, 5 years after that, what became really clear, because of the advances in molecular biology that had happened and were happening, was that engineers of all flavors, chemical especially, because chemical has a molecular emphasis to it, were being drawn in and recruited into the wonderful world of biology, because in part that's where the money was, but that's also where new exciting problems were. And like many engineers at the time, we started thinking about going in that direction, primarily to help biologists with their research, with their problems. So we jumped on board to help biologists study biology, usually by bringing in quantitative thinking and quantitative methods that they were kind of scared of. What I realized after sort of working with biologists for some years was that there was this fun road yet to be traveled, which I knew was going to be well traveled one day, which is, there's a bunch of neat little machines and parts and self-assembling parts. There's all kinds of stuff going on in biology that would make a toolkit for building stuff that might not have anything to do with biology. But as I started working with biologists, I came to realize, as the tinkerer in me, if you wish, as the engineer in me, that, oh, biology could work for me as an engineer. Okay. So that's the journey that I want to take you on today, is just show you how I sort of took the car around through the median, and I decided, okay, let me play around here. My intuition says this is a fertile playground. So it's not the classic story of I have an application in mind and I'm going to do what I need to do to advance the principles to achieve an application. Quite honestly, this is like, hey, this is like a neat playground. And what can I do with all these toys that I had discovered by being drawn into biology? So that's what the talk is going to be about, okay? And I'll gloss over some of the details because they're not obviously that important for this kind of a talk. So some parts of the slides I'll just sort of run past. Okay, so now how do you make that U-turn? Or in fact, how do you even drive down the road? So you know nothing about biology. I'm a chemical engineer. It's 1980, whatever, 1991, whatever. I don't know anything about biology. Where do you start? Okay, but as a chemical engineer, I do know a lot of things about thin films. And even just, even as an engineer, it's the package that's more important than what's in it 9 times out of 10 times, right? 9 out of 10 times, right? The water's not the big deal, but the sexy package that doesn't leak the water and so on, that's where a lot of the engineering comes in, right? So when I opened up my biology textbook, I said, okay, well, let's start from there and let's think about what are the packaging materials in biology. Maybe that's a point to start. I mean, I don't have to work with proteins necessarily or something like that. And of course, biology has fantastically interesting packaging material, and that's the material that holds the cell together. That's the cell membrane, right? So it not only holds the cell membrane together, this very thin molecular film, but it also is the packaging material which keeps the nuclear membrane together, and it keeps all this stuff inside the cell inside, and it keeps all the stuff outside the cell outside, except for stuff that it wants, like water, to go between. And what makes up the main ingredient of these cell membranes is, of course, something called a phospholipid, which has this split personality, the water-loving head group and water-hating tail groups. And in fact, they make this bilayer so that the watery inside of the cell is happy, and then the watery outside of the cell is happy because it only interacts with the water-loving head groups. So these phospholipids self-assemble into these cell membrane structures. Okay, and they're actually pretty interesting because not only do they self-assemble, but they are home, and they're beautiful homes for proteins and other kinds of inclusions. They keep certain things in and out at their discretion. And the most interesting part of this as a packaging material is that nature's not stingy. And you can just buy phospholipids. There's a lot of different phospholipids. They come in different flavors, if you wish. But you can buy these things and just essentially throw them in water. So you can just, for 10 cents, you can buy phospholipids and throw them in water. And what they'll do is they'll form essentially a bilayer package, shell. They'll form what we would call in this business a vesicle, with water on the inside and water on the outside. They'll just self-assemble into that form. So it's freeware. It's like, okay, I don't have to, see, I'm a chemical engineer, I don't want to make anything, I don't want to synthesize anything, I want to be able to just buy stuff, okay, and play with it. That's the difference between a chemical engineer and a chemist. I don't want to make anything, I don't want to be in the laboratory to make something, I want to be able to buy it. And you can buy these things, and it's even more fun than that, because not only can you buy phospholipids, and by the way, there's lots of different kinds of phospholipids, and that's a story within a story. You can throw them in water, they'll make these model membranes. You can easily put stuff on the inside. I'm kind of pooping out here. You can easily, configure it so that you put interesting things, soluble things on the inside. And it's not the only freeware that's available in the wonderful world of freeware for biology, because not only can you buy phospholipids off the shelf, but at the time when I got into this playground, you could also buy DNA off the shelf. You just say, make me a DNA, go to the web, make me a DNA molecule that's this long, that has these bases, and send it to me by tomorrow, and it would come in the mail by tomorrow. And of course, DNA assembles, and you can do fun things with DNA. So I realized, okay, this is the beginning of a really cool toy chest of stuff that I could buy. What could I make with it? What could I do with balls like this that could be fun, okay? And what's inspired me, well, one thing I'll just say, before as I said, I initially went into the world of biology by being drugged along by biologists who had interesting problems to solve. And we did what any chemical engineer would do. We used these to make model systems. We did some quantitative analysis. And if you really care, you can come up to me afterwards and you can ask me for very fundamental studies we did with biologists addressing biological problems. We worked on, for example, what is so cool or what is so interesting about the kinds of phospholipids that you actually, there's a specialized phospholipids that you find, for example, in human sperm. Those phospholipids, and they're a minor component, they do interesting things to the mechanical properties of the membrane that are unique to sperm, and you don't find these phospholipids anywhere else in the body. Actually, except for the brain, but I'm not going to go down there. But anyway, so there's all kinds of very, very interesting fundamental biological problems that we addressed using model membranes, using quantitative thinking. But again, what I'm going to talk to you about today is this playground. And it's inspired, it was inspired by what was, for me, you know, before phones and toys and Legos, by the Tinker Toy set, right? So when I grew up, your parents shoved this in front of you and said, build stuff, do something for 2 hours, okay? And what you got was you got balls and you got sticks, and you were supposed to like be creative and build stuff, okay? So I'm like, okay, so I know how to make balls, you know, these phospholipid membrane containers, vesicles. And in fact, let me think of DNA as the sticks, maybe, that I could use to tie the balls together. And maybe I could make something like that for some purpose as yet to be determined, to be quite honest, okay? So how could I do that? Okay, so I'll take you on a Highway Express journey of all the things we have been able to build with components that I could buy. So as long as I could go to the web and buy it, order it from Aldrich or order it from a We'll Make You a DNA, I did it. I didn't want to synthesize anything. What kinds of cool things can I make, all right, using these DNA parts, using these biological parts? So that's the premise of the talk. What can I make? And then as we make these, we're trying to think what we could use them for. All right, and as the talk proceeds, I'll take you to more and more levels of complexity. And just in case you want to know where we are in the talk, I'll show 6 different levels of complexity. So if you kind of zoom out on level 2 and you see with level 4, that just means we're at a higher level structure. Okay, so that's your little ticker for where we are in the talk. Okay, so here we go. All right, so I've got balls and I've got sticks. Whoops, what happened there? It's moving on the computer, but it's not moving. Okay, there we go. Okay. All right, so I can make these balls, I can make these vesicles in different sizes. That's easy. You can make nano, you can make micro, you can make macro vesicles. You can really make these vesicles in different sizes, called large or giant, and then I've got DNA. Okay, so now, let's see. Okay, I got to press here. Sorry, this is a, okay. So the DNA is going to be cool as a part of the Tinker Toy set because you all get the DNA thing, right? You've got the matching strands, and they'll hybridize together if you get hybridized strands. And the cool thing about DNA as a glue, as an engineering material, is that I can unglue it just by raising the temperature above the hybridization temperature. So that's like the cool thing from an engineering perspective about DNA is you raise the temperature, you can unglue something. So as a molecular glue, you have a way of gluing and ungluing. So the only trick there is how do I use the DNA as sticks to glue together the balls? Okay, well, that's not too hard of a trick. In fact, that was sort of already out there in the literature. I didn't have to invent it. Again, it's pretty easy to just buy DNA and you tell the manufacturer, stick a cholesterol molecule at the end of the DNA. Okay, so cholesterol likes to live in cell membranes. Actually, cholesterol plays a minor supporting role in the whole talk, in various different actors in this talk. But in this case, cholesterol is just a hydrophobic molecule which loves to burrow itself in a membrane, and you can buy DNA with cholesterol attached to one end, okay? So now I've got a way to get the sticks inside the balls, and you can put one kind of stick in some membranes and the mating stick in the other membrane, and in theory, they should tie the balls together, okay? Well, it turns out, I'm going to jump ahead, because it turns out that with one cholesterol foot in the membrane, it kind of pops out. That DNA will kind of just pop out. It doesn't stay so well. So there's an advanced trick you can use. I'll just show it to you for those that are really interested in the details. The advanced trick is you make a little more interesting. You see, you have one DNA strand that's like long, and the other one that's a little bit shorter, so that you can get 2 cholesterol feet in. That still leaves you a part of the DNA that you can hybridize with a mate, but then you can actually get that stick to stick into the ball a little better. It's just a detail. But the point is that it works. So the idea is you should be able to create DNA balls with sticky rods coming out that will find and hybridize with their mates below a certain temperature. And it should be reversible. If you raise the temperature, you should be able to have the balls drift off, and if you lower the temperature, the balls should all coalesce, and that's exactly what happens. So let me show you a quick picture. So you form vesicles, and these are all in the collapsed state, so all the DNA is hybridized, and all the balls have come together to form like a cloud. And then you raise the temperature. All the motion is because it's just done at fast speed in an oven. But you raise the temperature, and the balls all disperse, and then the balls will come back together, and the balls will disperse, and the balls will come back together, and you can just keep going until you're bored, okay? So that's really cool. So you have this way of gluing and ungluing with temperature these balls, these vesicles, these lipid balls, okay? So that's not the only lever you can play with when it comes to DNA. DNA will not only hybridize or unhybridize as the temperature changes, but if you monkey with the salt concentration, that will also hybridize or unhybridize DNA. So what we found is we could create different regimes. So for example, if we don't have a lot of DNA sticking out of the balls, they weren't going to be very adherent. If we had a lot of DNA shrouding the balls, they would be very sticky. And we found different domains, if you wish, where we either had no aggregation between the balls. I'll tell you what the red and the green is in a minute. Where we had no aggregation or small clusters or really large clusters. And that would depend on the amount of DNA that we shrouded the balls with, or it would depend on the salt concentration. But the nice thing about the salt concentration is we can change that in situ. So we could basically take a system that was collapsed basically at one salt concentration and basically uncollapse it or collapse it just by changing the salt concentration. So the point is there was just a lot of flexibility here in creating these clusters. The problem is the fidelity of the system wasn't that good, because the green is supposed to be one DNA, the red is supposed to be another DNA. So you should only see binding between green and red, not between green and green and red and red. But what's happening is those DNA sticks are kind of jumping out of one vesicle and jumping into the other vesicle, and it's kind of mixing the whole thing up in a way. But the principle was there, and so we were happy that we could at least control, to some degree, aggregation and unaggregation. Okay, so that was the simplest and the first thing we did. And as I said, you could go from aggregated to unaggregated states just by basically changing the salt concentration. You have something aggregated, move it into a slightly different salt concentration, and all of a sudden it would disperse. Okay, so temperature, salt. Okay, so that was the first thing. All right, what did we do next? Well, just with Tinker Toys, when I was 2, the Tinker Toy sets were made out of wood. But when I became 4, my parents brought home a new Tinker Toy set, okay? And it had something to do with the movie The Graduate, which you probably have never seen, and a classic line in The Graduate, which was, who knows? Plastics, okay? Right. So now I'm, 4, 6, whatever age that was. I come home, and now it's a world of plastic, okay? Okay, sturdier, okay? Hardier toys, plastic. Well, there's an analog in the world of membranes. So not only do natural phospholipids make these beautiful membranes, But polymer chemists around the world and in companies were making polymers that had exactly the same hydrophobic head, hydrophilic head, hydrophobic tails. And they found that when they threw the polymers into water, the polymers would form the same balls, the same kind of vesicles that the phospholipids were. So they just made polymeric analogs to phospholipids, but they're just much longer molecules. So the same stuff should work. Well, first of all, are those polymer balls tougher than the phospholipid balls? Yes, they are. So again, the polymers just has a bigger head group and a bigger tail group, but they'll do the same thing that the phospholipids do. They'll form vesicles when you throw them in a solution. And if you ask about the mechanical properties of these vesicles, or these polymersomes, instead of vesicles or liposomes, vesicles made of lipids, these are polymersomes. If you basically tug on them and try to stretch them and break them, and you can do this using what's called a micropipette technique, they are indeed tougher. What's interesting about these polymersomes is they have the same elasticity, if you wish, of the phospholipid membranes. These four-letter combinations all refer to a particular kind of lipid, so just don't worry about it. Those are lipids. These longer things are polymersomes. But same elasticity, but much tougher. You can really stretch them much harder until they pop, okay? Polymers, tougher, okay? So how do we get the DNA stick to work with the polymersomes? Well, that one you can even give a high school student. Do you think the DNA I was using before will work? No, because it's not long enough to stick out of the polymersome. So in fact, all you got to do, so here's the polymer membrane, or at least one half of it. If you use the little DNA stick that we used before, it's not long enough. It buries itself inside the head group or the tail group. All you got to do, you can ask a 7th grader to crack this problem, is just order basically a spacer between the cholesterol and the actual DNA, and just make sure the DNA sticks out. That's all you got to do. So now it's kind of fun, because not only can you link polymer balls to polymer balls using the DNA, but you can link polymer balls to your smaller liposome balls. No problem, right? So you can actually make superstructures like this, where you have a polymer ball, and now you have lipid balls, smaller lipid balls. And that gets kind of interesting now, because now you could think maybe one day of using this as a drug delivery device, okay? Or maybe you put maybe this becomes the carrier somehow, and maybe you put different drugs in these balls or whatever. But the key thing is you can easily destroy the lipid balls and release their contents without having to destroy the polymer ball. Again, we weren't so focused on applications. We were focusing on the fun things we could make. All right, so that was the second thing we were able to do. And just ignore that. I'm going to skip past that. Okay, but now we wanted to raise the stakes a little further. And what we realized is that we had an opportunity to capitalize on one of the coolest things about lipids, okay, phospholipids. So even just like in 3 dimensions with any material, it undergoes phase transitions, solid, liquid, gas, right? Well, these phospholipids, in their world, in the 2-dimensional world that a phospholipid lives in, they have their own phase transitions. And again, in the world of biology, this was becoming a really cool thing about cell membranes. The biologists were realizing that the phospholipid backbone of a cell membrane itself was in different states, liquid-like states, solid-like or gel-like states, something called a liquid-ordered state. It's like somewhere in between. And you had to mix as with much phase behavior in 3-dimensional world, the more components you add to the system, the richer the phase behavior gets. So for example, when you would mix different things together, a lipid, another lipid, and that minor actor that I told you appears in biology a lot, cholesterol, together, you could have in the membrane itself a variety of different phases. So this is a ternary phase diagram of all the different phases that might exist. There's liquid ordered and disordered. And so the membrane itself would be textured, self-textured. So we thought, now we have anisotropic balls, right? So the ball itself, the lipid ball, is not homogeneous. It's inhomogeneous. It's got spots or dots or whatever. And now maybe we could make the dots or the spots selectively sticky. Okay, wouldn't that be cool? And since cholesterol is involved in this somehow, so some of these dots are cholesterol rich, the liquid ordered, maybe we were hoping, oh, maybe we'll get lucky. Maybe when we add our cholesterol sticks, our cholesterol tagged DNA, maybe we'll be lucky and it'll go like all into the phase that's a little more cholesterol rich. And then we'd have sticky, selectively sticky balls. Well, we weren't that lucky. All right, so we added cholesterol, we added our sticks, and they kind of went, everywhere, okay? We had just as much cholesterol DNA in this liquid disordered phase as we had in the liquid ordered phase. So it's, I think, 1 to 1 when we measured it. So then we worked really hard and we said, well, man, what about that two-footed cholesterol? Maybe that will selectively go to one of these two phases. And it did better, but about 2 to 1. Okay, but so we weren't able to make anisotropic balls that were selectively sticky because both sides were sticky with this DNA. But now we were able to call upon our intuition from driving down that other side of the road. Because remember, a lot of the work we were doing at the same time was we were trying to help biologists understand biological problems. And we were working with a couple of life scientists who were very interested in mitochondrial membranes that have a lot of cardiolipin in them. And cardiolipin is this really funky specialized lipid. And we had reason to believe, and I won't go into the whys or the wheres, but we had reason to believe that if we added some cardiolipin, this funky lipid, to this system, we might actually be able to get our DNA-footed, our cholesterol-footed DNA to go into one of the phases, and we were right. So we did that. We added cardiolipin to the system, and now we were essentially able to get truly selective incorporation of our cholesterol-footed DNA into basically just one side of the ball, or one phase of the two phases that existed. So now we were good because we could make anisotropic, selectively sticky anisotropic balls, and we could play with those. And now when we had that capability, now we were really able to start making much more interesting structures where really it was like the green part had this sticky part to it and the red part didn't, and now we can make all kinds of funky structures much more on command and with much more fidelity because we were able to make these selectively sticky balls. So this is just some data that's showing you it's not fusion, it's just stickiness. But the point is in the pictures, okay? So we were able to make some really cool things. Okay, so that's number 3. All right, so then we're thinking, okay, well now life's getting kind of interesting because we started to think about balls that were inhomogeneous. And then we were thinking, we've been playing with these polymer balls, and they're really tough and they're really nice, but proteins are never going to want to live in the polymer balls, these polymersomes. Proteins want to live in these lipid balls, liposomes. And we thought maybe we could make polymersomes. Wouldn't it be cool if we could make like a lipopolymersome? Could we possibly make a polymer ball that had like a lipid window? Wouldn't that be cool? It's a different form of anisotropy. Because then, if we could do that, then we could start adding sticky DNA to either the polymer or the liposome. But we could also ultimately add proteins and more biological freeware to the lipid window in the polymer bubble, okay? So how do you do that? Well, we did like you try the most obvious thing. Why don't we just mix lipid and polymer, throw them in water, and hope it works? And it did. So what we found was, and it kind of depended on the lipid, okay, it depended on the polymer, but this is a lipid that actually likes to make very solid-like lipid phases when it's on its own at a certain temperature. So it's fluid-like, and then if you cool this lipid, it'll make a very rigid membrane. So when we had it at a higher temperature, we mixed this particular lipid with this polymer, and basically at a high enough temperature where this thing was sort of more fluid-like, and it was a nice dispersion. The lipid and the polymer happily coexisted. But as we cooled below the natural gel or phase transition temperature of this lipid, it started creating lipid windows for us. So we cooled it, and then all of a sudden the lipid would aggregate, phase separate, if you wish, into a lipid window. Pretty simple. And in fact, it was more generalizable than we even imagined. You didn't have to use this particular lipid, which has a very strong phase separating characteristic. It forms these gel-like, harsh lipid phases. It turns out it worked pretty much with almost anything we tried. So it worked with, remember that liquid ordered phase that had cholesterol in it? It worked with that. And then you even got round windows, so that was cool. And then it even worked with the most prevalent lipid you have in your cell membranes. There's a called POPC. That's like the major lipid in there. And it even worked with that. So boom, you could create these wonderful windows. Okay, so this polymersome, and then this prevalent lipid, and you have to add a little cholesterol, and boom, makes these lipid windows. And then what was cool is, depending on the rate at which you change temperature, you can change the size and the number of the windows. So if you cool really fast, you make a lot of small windows. If you cool really slowly, you make big windows. Okay. So for example, here's different cooling rates for that original lipid that I showed you. It makes a big window or a bunch of smaller windows, and you get the idea. Let me show you a couple more pictures. What's particularly cool about this picture, and this is the reason why I'm showing it, is sometimes when you make these lipid balls, you inevitably capture smaller lipid balls within it. So this big ball, it's polymer and lipid, this big ball has a smaller ball inside of it, and it's showing you that it's happening in all the vesicles, in all the polymersomes at the same time, because as you cool, you're getting windows on the inside ball, and you're getting windows on the outside ball. Okay, so we can do that. So now it gets more fun. It gets even more fun, because now, if you've got a window, first thing you want to do is you want to open it and close it, or you want to break it. So now we can do that. So, no, okay, before you open and close it, you can obviously make the window selectively sticky. We know how to do that. We can make the lipid part sticky, we can make the polymer part sticky, and then we can ask the windows to stick to one another. Easy. We've done that before, essentially. Or we knew how to close the windows. So it turns out, now remember, the lipid in this particular assembly, polymer, these are the lipid windows, has POPC, that's a lipid, and cholesterol in it. Okay, so you need that cholesterol. I told you, cholesterol is a minor actor in the whole story here. It turns out that this molecule called cyclodextran is a vacuum cleaner for cholesterol. You throw cyclodextran in the system, in the water, and it basically, the cholesterol that's part of this just would rather be in the middle of that. So it leaches the cholesterol out of these membranes. And as it leaches the cholesterol out of these membranes, the lipid doesn't want to be phase separated anymore because it needs the cholesterol to be phase separated. So the lipid slowly goes back into the polymer matrix. So it's just a way of gently closing the windows. Okay? So if you create windows, you can close the windows. You can smash the windows using other kinds of molecules. So if you throw a soap, this is nothing more than a soap, okay, in the solution, that'll actually just destroy the lipid windows. Or if you throw in a biological molecule which bites the head off of the lipids, that'll destroy the lipid windows. And now this is a violent process. So it's really like taking a brick and throwing it against the window. And this is a way to control release. So we can, ooh, is that me? Is that a sign? Should I go faster? Okay. Oh, ooh, okay. Oh, I guess they were trying to make so you could see the window. Okay, so I think you can see the crashing of the window here. Let's see. See the window being broken? So you just throw in the surfactant, and boom, the window is just broken, and the thing closes upon itself. And what's cool about that is that let's go to here. Is that you can use this technique of breaking windows to control release. So this is just an experiment where we filled the vesicle with something, this green dye. We broke the window using that smashing technique that I showed you, and now there's no more green dye left. So you can actually control release by breaking these windows. Okay. Okay, so that was another cool thing we could do. All right, so then what did we decide to do next? Oh, sorry, one more thing. Turns out when you start making these balls with windows. There's all kinds of neat design things that crop up, like bow windows and peanuts, and you can start to play with different things like the line tension and the osmotic pressure, and you can create all kinds of funky structures. Just a commercial break for that. Okay, it's a really rich playground. Okay, so 2 more examples of fun things. So when we got to this point, had a new postdoc join the group, and we were trying to think about what's really the next level of complexity we could go. And I had just come back from a trip to Minnesota, my PhD alma mater. Well, why did I come back? Well, it's when I was a graduate student, I saw a sign on a telephone pole that said, walk on a treadmill, come get 20 bucks. Who doesn't need 20 bucks? Okay? So I go over to the med school to walk on a treadmill for $20. And what I realized is they were starting at the time what they hoped to be a continual running study of the progression of heart disease in young Americans. So 4 universities had written an NIH grant, Minnesota, someplace in San Diego, Alabama, whatever. They wanted to get a bunch of 20-year-olds. They wanted to get 8,500 20-year-olds and monitor them through life as they would and get the data needed for the progression of heart disease. So this was a study called Cardia. And in fact, it's still going today. I just went back the other summer for the 35th or the 40th or whatever. By the way, just the amusing thing about this, the not so amusing thing about this, if you look at our weight as a function of time from when we are 20 to when we are all now like pushing 60, it is a line straight up. Okay. And there's more scary things. But anyway, so I had come back from this, and about the same time, the postdoc in the group was independently trying to think about new directions we could take the research, and he realized that the, LDL and there's no bad and good cholesterol, right? These are carriers for cholesterol. Like cholesterol is just a molecule. It's neither bad or it's good, right? What makes a cholesterol, the good or bad cholesterol is associated with the carriers of cholesterol. And what they look like, basically, is this. So what this is it's a protein belt, and there's just 2 of them. Okay, they're just different colors so that you can see them. It's a little protein belt with a little phospholipid membrane, like a pancake, with the belt around the side of the pancake. You get it? All right? So it's a little phospholipid, and the cholesterol buries itself in the phospholipid membrane. So that's the carrier. That's what's shuttling cholesterol back and forth in your body, is a little nanomembrane, a little chunk of a membrane, and instead of being a ball, it's a little chunk, and it's just got these 2 protein belts around it. So we said, oh, cool. All we got to do is get our hands on these protein belts, or a synthetic version of these protein belts, and we knew somebody at Illinois who would give us some. And why don't we try to make these membrane pancakes? Because then we could put our DNA in the pancake instead of the cholesterol, and now we could start linking the pancakes together. Wouldn't that be cool? Maybe that would work, and it does. It worked beautifully. So this is just some details on what those pancakes look like, but they're small. Now we're talking about nanometer level stuff. Now you really got to use heavy-duty electron microscopy to see what's going on. But the idea was we would try to get our cholesterol-footed DNA in the pancakes, and they should assemble or disassemble with temperature, just like the balls did. And it works perfectly. I'm going to have to speed up, guys. So let me just show you that under the microscope, when you look under the microscope, this is exactly what you see. You see these columnar structures that are strung together. And the cool thing about these is there's actually a his tag, like a belt buckle on the belts, where you can attach little metal balls if you want to. So not only can you make these columnar structures of these lipid pancakes, but you can even decorate those columnar structures with metals, and then ultimately maybe make a wire if you wanted to put enough effort into it, a nanowire, by basically, I'm running out of time, so I'm going to speed up a little bit, by basically adding metal balls to the belt buckles. Okay. So we got this far, and we said, well, this is really cool, and if we really wanted to spend a lot of time on this, we could really start to optimize this process to make these wires. But instead, we pushed on to an even higher level of complexity. So this is where I'll end. And what's the inspiration there? Okay, well, it's like we all crave motion, I mean, you can make the balls or you can make the pancakes, but we really wanted I don't know, we just had this, how do you get more animation into these structures? Could we possibly attach these balls to something that would actually move? Something maybe that had a membrane, that we could use our membrane attachment chemistries and attach it to other membranes that would actually move, that would do something, that would take our little balls somewhere and maybe drop them off. So we had done work on sperm. Remember the comment I made a half hour ago? And so there's a single-celled membrane that moves, okay? And we said, well, wouldn't it be cool if we could attach our little vesicles, like little Samsonite bags, to the sperm and let the sperm take them where they were going to go, and maybe drop them off during fertilization? Wouldn't that be cool? And then they could drop off stuff that we wanted dropped off during the fertilization process. And so we didn't really have to develop a lot of tricks to get this to work. We didn't even have to use special DNA or sticky glue to get vesicles to attach to sperm. We just had to come up with the right, in fact, it worked almost too easily. Positively charged vesicles would just naturally attach to sperm. Negatively charged vesicles would attach to sperm. There was aggregation issues associated with that, but through a clever choice of phospholipids, and again, because we had been working on these fundamental problems with membranes, we sort of had an intuition about what to pick. But basically, we could make vesicles that would attach to sperm. And interestingly, the head loves to collect itself with vesicles, okay? And the cool thing about it is that the sperm don't care. They're just not bothered, okay? So all the things that sperm do that you track, like if you're a fertility doctor, you track motility, you track the speed, you track the this or that. With or without all these vesicles all over them, like they don't care. They just don't care, okay? They go just as fast, they beat just as speedy, they just don't care, okay? And a lot of biological assays that are important or that we could run, they didn't care. They had these. So for example, in the fertility clinic at Yale, in collaboration with whom we did this work, there's a particular binding assay that they use to check the health of sperm. They're supposed to bind, the heads stick like crazy to this particular acid that's on a slide, and they try to get off, but they can't. And that's a good sign if you're a fertility doctor, or if you're giving sperm, you want that to happen. And even if our sperm were shrouded by the balls, the vesicles, they behaved just like that. It didn't matter. So we said, well, this is really cool. Well, what about the ultimate test? Will they fertilize? So we worked with our friends at Yale, and of course you can't do this with like human sperm. It's like illegal, okay? So you have to use mice, okay? What's really cool about mice is their sperm have like super long tails. I guess it has nothing to do with mice, but they have like, their sperm is like super long tails. So it is. So that's a human sperm, and that's mice sperm, super long tails. But they just take on these vesicles, no problem. And I won't go through all the details because I am running late. But basically, there's a marker that doctors look at to see if fertilization has happened. There's something called a bipolar, two polar bodies. And if it happens, you know you've got successful fertilization. And again, I don't have time to take you through the details. But the punchline is it works. So we can basically get our hybrids, the sperm coated with the baggage, to fertilize cells, okay? And therefore, we can have the sperm deliver things to the cell during fertilization. So this is just, the next slide just gives you a quick example where we delivered green fluorescent probe to a cell. So, you know, the postdoc who was working on this went back to Europe, and then we were at the point where we were just handed this off to the doctors at this point. And, we patented it, and you can play with it. But ultimately, this may end up being the killer application that ever comes out of this work, is a way to, a novel way to bring material into cells during fertilization. So they're working on it. We're done with it at that point. And what we did is we directed our attention, and this is the last thing we've been doing in the lab, to other single-celled things which have a membrane package themselves, and that's bacteria. So the latest thing we've been doing is trying to play with attaching vesicles to bacteria. Okay? So now you can imagine, well, maybe a bacteria in a soil could take our vesicles and maybe drop them off in a remediation process or whatever, okay? And again, I don't have a lot of time to go into this, and this is the very latest stuff we've been doing, we've been playing around with. But the cool thing about, so it works, and you just have to be clever about which lipids you choose. You can either attach, the cool thing about bacteria is they're small too. So you can either attach small vesicles to bacteria, or you can attach bacteria to big vesicles for whatever reason. So now you can actually play with all kinds of funky size changes. And again, you can attach vesicles to small or small to big or whatever. You can have all kinds of fun here. So we're playing around with this in the lab as we speak. But my time is up. I think I spent too much time. So let me just thank these folks who've been with me over all these years, just one at a time, sort of carrying this work on from one level of complexity to the next level of complexity to the next level of complexity. And most of all, let me thank you for your wonderful attention and this wonderful visit to speak to you today. And I'm very much looking forward to seeing all your posters in a couple of minutes. So thank you. Some lights?
Do we have time for questions of any sort?
Were you able to tell if a sperm is carrying like a vesicle and brings it into an egg, if the egg makes use of whatever is inside the vesicle?
We never took it to that length. We just stopped where we showed that, for example, it could bring a green fluorescent probe in and it could be expressed. But we didn't do anything more complicated than that. Again, it was, in scientific research, it's not so much lack of wanting to work on a project. It's usually people. People move on with their careers, and the postdoc that I had who was working on that had to go back to Europe to continue with her life. And so a lot of these projects you just sort of leave at different stages for reasons far other than your inquisitiveness on it. Good question. Sure.
I was wondering, since you can obviously decide what sequence your oligos are going to be on the surface of the vesicles, are there situations that either you've done or you've been interested in doing where you would try to selectively have different sequences in different places so that you could assemble particular shapes out of the vesicles, sort of build a structure by selectively varying the sequences?
Yep, so he's asking about all the degrees of freedom you have, because you can take this DNA, you can build it however you want with different kinds of sequences, different kinds of lengths, and then you could really go to town. The only thing that we did that took advantage of the ability to monkey with sequences is when we were playing with those pancake columnar stacks, we wanted to prove that if we got longer DNA, the spacing between the stacks would be that much longer, and it exactly was that. So we could control to within base pairs the spacing between those pancake stacks. But we never went crazy. And again, so many of these projects have very natural parameter spaces where you could burn 5 PhD students if you wanted to. Well, you look at this, and you look at this, and you look at this, and you look at this. And I think one of the hardest things about research, maybe to bring it back to sort of the general topic, is how do you know, because where do you move? Up, down, left, right? A lot of it's related to the people you have in your lab. A lot of it's related to their interests. When you're building a career, is that kind of work, interesting as it may be, going to be career defining and it's going to separate you out, and is that going to be your tenure case when you do it as an assistant professor? So it's got to be important enough and interesting enough, not just fun or of interest. And so I think those are the big questions that you wrestle with when you think about research as a career. How does this all fit together in terms of personnel, mentorship, impact, your career? Because you're never going to be short of problems you could look at. You're never going to be short of ideas or things that would be fun to do. That's never the problem. The problem is, what do you do and with whom do you do it? And maybe that's probably the most valuable thing I might have said today. I don't know.
Well, that's a good place to end.
Perhaps.
Thank Kyle one more time.
Thank you.
Now, after our cliffhanger earlier today, we will continue. I'd like to introduce Professor Dean Wilcox to announce the winners of the Sigma Xi Christopher G. Reed competition. After the presentation of those awards, I would like all of the WISP interns to gather at the front of the room for some photographs, and then please circulate throughout the building to see all of the research that the undergraduates have done this year.
So I understand that our delayed contemplation of the competition led to some anxious moments, but here we are. Just to say a few words about what is Sigma Xi. Sigma Xi is an honor society that was started in 1886 by engineering students and faculty at Cornell University. So this society actually grows from the engineers. It's grown to become an international organization to recognize and honor and promote excellence in science and engineering, and is now broadened to include many allied areas: education, philosophy, ethics. And membership worldwide is 80,000, over 500 chapters worldwide. In fact, most Nobel Prize winners have been a member of Sigma Xi. The organization publishes a very award-winning magazine, American Scientist, very accessible science articles, and also provides small grants for funding for individuals early in their career in particular. Membership in Sigma Xi, which recognizes excellence, is at two levels. One is at the associate level for individuals who are early in their career that have the promise for excellence, and at the full level for those who have distinguished themselves as practicing scientists. The Dartmouth chapter of Sigma Xi has two main events every year. One is to induct new members, and the second is the Christopher Reed Science Competition that we've held now for many years in partnership with WISP and the Science Symposium, and we're grateful for this forum. This is a competition among students who've done honors thesis research in the sciences, and it's judged by a panel of members of the Sigma Xi officers of the chapter. Chuck Dallian, is Chuck here? Everyone knows Chuck. Chuck runs the electron microscope lab. You know, Chuck knew most of the participants because they brought their samples to him. Robin Barbato, who works at Krell, not here. Natasha Grotz was a member of the judging committee this year. And finally, Tim Smith from the physics department. So this is a panel of judges. And I'd like to say that we were late in deciding the winners because there were so many participants this year, a record number of the members of the class of 2017. And it was tough to decide on winners. It is every year because it's excellent science, it's outstanding science. And as recognition of that, all the participants will in fact be inducted as associate members of Sigma Xi. But we did have to decide on some winners in this competition. So let me start with the third place prize winner. For his research in earth sciences under the advice of Professor Justin Strauss, geochemical constraints on middle Paleozoic magmatism and the northeastern Brooks Range of Alaska and Yukon Territory, Canada, Josh Perez. We have 2 second prize winners. The first, they're equal, second prize. The first of these, for his research in engineering, under the advice of John Zhang, the thesis titled Circulating Tumor DNA Screening and Capture Chemistry for Pancreatic Cancer Detection, Yichen Zhang. Our other second place winner, for his research in computer science, advisor was Emily Cooper. The title, Helping the Visually Impaired Navigate Using Text Spotting, a HoloLens application, Jonathan Wang. There it is. Normally, we try to reserve a single first place prize, but this year we couldn't contain ourselves. There were 2 that were just so outstanding, we had to give 2 first place prizes. So the first of these, for her research in engineering under the advice of Ulrike Best, structure property processing correlations in freeze-cast collagen scaffolds for biomedical applications, Isabella Caruso. She's already back in the lab. Yes. Okay. The other first place winner for her research in the biology department, advisor of Eric Griffin, title of A Comparison of Maternal and Paternal Mitochondrial Dynamics in 7 Species of Nematode, Mallory Rutigliano. And these winners will put their ribbons up by their posters, so please stop by and congratulate them. Congratulate all the participants, because it's just an amazing work that we have to judge every year. So, thank you.